I'd like to pose a few questions to you. My background is in the laundering of textiles, and how important is laundering in healthcare? Media attention. If you look at the TV reports, the daily newspapers, you see things about car parking, high charges. We look at the cost of food. It costs more to feed a prisoner than it does patients in hospital. Dirty hospitals. The number of people that were affected by infections that have been contracted from dirty hospitals. Papers full of it. Nothing about linen. Nothing that is unless you really start to dig deep. 1995, there's an occasion where babies at birth find all free of any bacteria, but within a day or two found to have infectious. It was finally traced back to baby vests that were laundered on site in domestic washing machines, domestic type tumblers, and was causing quite serious problems. The problem's been eradicated simply by autoclaving all baby items. 5 children contracted infections in the States. The reason it takes so long to come out, this is still the subject of a court case. 2 children died as a result of an infection caught that was again traced back to the fact that staff clean linen on a loading bay was being sorted alongside medical waste being stored. The same trolleys were being used to transport clean linen and soiled linen. Linen kills again. Hong Kong this time. Patients died as a result of contracting infection that again was traced back to the supplier, a commercial laundry supplying 14 hospitals in Hong Kong, where on inspection 50% of the linen was contaminated. That's 50% of its supposedly clean linen contained infections. Do textiles used in healthcare present a risk? I'm just a humble laundry guy. Not sure. The experts will follow. Professor Sally Bloomsfield, she talks about laundry being not only visually clean, but hygienically clean. And there's a big difference between looking clean and actually clean. We all go to the toilet, our hands look clean, but we still wash them when we come out. Dr. Lisa Hackley, she talks about if you work with a food processing factory and you wash your uniform with the family's wash, that could include underwear, it could become infected with E. coli or salmonella or whatever else is on those items. The facts, just to give you an idea of how big an issue it could be. 94% of healthcare linen in this country is processed by the private sector. 91% of this is processed by 2 companies. One that used to be owned by a German, now it's owned by the French, and the second is financed by the Belgians. Linen used today in St Thomas's Hospital down the road could be used in a hospital in Oxford tomorrow. Laundry goes everywhere. 521 million items a year are used by the NHS. 177 million pounds. It's probably the oldest profession in the world, or so I thought. Someone once said, well, medicine's older than laundering. No, it's not. I got off a train last night at King's Cross, and I don't even believe that it's the oldest anymore now. It's simple. It only becomes complicated when it's a business. Uses basic technology, Mechanical action, we shake it, we rub it, we wipe it. Water holds the soiling, carries it away. Soap, detergent, helps mechanical action, suspends soiling. Heat, a catalyst to allow the chemicals to do the job and also to kill bacteria if sufficient heat is applied. And time, sufficient to do the job. So what's changed? Most linen used in healthcare is processed to an high standard. HGMO 104, the new guidelines published by the Department of Health, it replaced the old Laundry Manager's Bible, HSG 9518. It replaced a 12-page document with one of 161 pages, which probably accounts for very few people reading it. We now have EN 14065, a European standard to which all healthcare laundry is subject to. I say all healthcare laundry, it excludes mops, which is probably the single most highly contaminated item there is in a hospital. It's not subject to any conditions. Wash them, rinse them out, do whatever you want. No standards. And it also excludes uniforms, which can be washed at home. Is there a risk? 2007, the Department of Health undertook a review on uniforms to see, is there a risk of staff washing uniforms at home causing a risk? A guidance document was published on washing items at home. Quoted in the document, it said there is little effective difference between domestic and commercial laundering in terms of removing microorganisms. That's something I would dispute. All beds are changed with fresh linen after a patient is discharged. Curtains are only changed 6 monthly, or if soiled. Do they not present a risk? Uniforms washed at home, they are not considered a risk, but are they? Is there a cause for concern? Our next speaker hopefully will demonstrate that. Lisa?
Good afternoon, I'm Katie Laird. I'm a microbiologist and I'm head of the Infectious Disease Research Group at De Montfort University. And I've got quite a keen interest in textiles, but not only textiles, what's happening in our textiles in hospitals. This presentation mainly focuses on nurses' uniforms. However, there is also some data in here on hospital sheets. So, we're going to look at the survival of microorganisms just on textiles, so on polyester and cotton. We've had a look at the NHS policies on home laundering of uniforms and whether these are actually adhered to. We've looked at the effect of low temperature laundering on microorganisms, and we've looked at what risks this potentially poses. So, first thing that we wanted to establish is, right, do microbes survive on textiles? And if they do, how long do they survive for? There is published literature out there, but we wanted to look at it back down to the fibre rather than the textile as a whole to understand what nutrients availability, etc. was there. So we chose E. coli and we chose Staphylococcus aureus, so we had a gram-negative and a gram-positive, and we used polyester and cotton fibres. As you can see from this figure, if you look at the top two lines, that is cotton. So that's the green for the staph and the blue for E. coli. And it's still there in considerable numbers at 21 days. When you look at the polyester, there is a greater reduction much sooner, after about 7 days. So what we believe is enabling the microorganisms to survive on the cotton is both moisture and some nutrients availability. So we know that microorganisms can survive for significant amounts of time on these fibres, so on uniforms, on bed sheets, before they are laundered. And then we thought we'd just have a little look at them and see how they looked, what they were doing. And we looked at them over a period of time. So we looked at them at 8, 24, 168 hours, which is 7 days, and 504 hours, which is your 21 days. On the polyester, you can just see scattered microorganisms on the surface. However, when you start looking at the cotton fibres, for both the E. coli and the polyester, you're seeing clumping of microorganisms within the fibre. You would expect this because a polyester fibre is very smooth, whereas a cotton fibre has got more threads fibres coming off of it, and places for microorganisms to attach to. So not only were we getting viable counts back, we're actually able to see that the microorganisms are surviving on these textiles. So what's the risk? Firstly, if domestic laundering of uniforms does not occur straight after the shift, and those uniforms are sitting in your laundry basket at home, then the microbes are still viable. Have you put other items of clothing in that laundry basket? Are they coming in contact with the side, the chair, wherever you've thrown your clothes off after a shift? What's happening? Where are they being transported to? And those cross-contamination sites within the home is not something we've looked at, but there has been studies that have looked at contact points on washing machines where you're using the handle to put the uniform in, etc. So the fact that the microorganisms are able to survive and are there is a potential risk. So Ian touched on what the policy said, but what does the policy say about the uniforms themselves and what nurses should be doing with them? The policy states if you wash at 60 for 10 minutes, it removes almost all microorganisms. However, washing at lower temperatures using detergent eliminates MRSA and most other microorganisms. There's no numbers in there, there's almost and most, and as a scientist, I'd quite like to know what the actual numbers are and what potential risks they pose. So what we did was we sent questionnaires out to 4 trusts, and we just asked the nurses, how do you launder your uniforms? How often do they get changed? How many shifts do you wear them for? Do you use detergent? Do you tumble dry? Just to look at the practice of what was actually happening in reality. 3 out of 4 of the trust policies, when we looked at them, stated that there was no requirement for detergent, which is slightly worrying. And 1 out of 4 stated a minimum temperature of 50. So we're talking about a lower temperature there. And what the nurses said they were doing was mainly washing at 40 degrees Celsius and 60 degrees Celsius. There was a 38% against 45%, something like that. We did have people hand washing their uniforms, which was of concern. But the policy states 60 for 10. Now, the majority of nurses were doing that. However, what's happening at that 40 at 10? What's happening to the rest of those uniforms? We also split this down into infectious disease wards and non-infectious disease wards. We classed A&E as infectious because you're not sure what's coming into that department. Because we wondered if there was more knowledge and training around those that worked within an infectious disease ward. We didn't see any significant difference between the way we analysed the data. The other thing that should be stated is that the uniforms should be washed on their own, so not in with babies' clothes or children's clothes or a vulnerable elderly adult's clothes. Again, around 60% of nurses were washing there alone, but the other 40% were washing them with other items of clothing. And the data I'm about to show you will show you what potentially the risk of doing this is. We took the 40 and the 60 degrees washes, and we used biological detergent, because that was the detergent that the nurses stated, that, or the healthcare workers stated, that they used most. We contaminated some fabric A is 65% polyester, 35% cotton, which is the most common uniform makeup. And we also tested 100% polyester. We inoculated it with E. coli and then another samples with Staph aureus, and we laundered at 40 and 60 just to see what happened. We did use high loads, maybe higher loads than we picked up in the hospital environment, but this was so that we could calculate the reduction. And what we found was that if you washed at 40 degrees Celsius, that around 4 logs of bacteria were remaining on the uniforms or the textile swatch, and that also around 3 logs of that was being transferred over to sterile cloth that was in the wash. So there was a transfer of those microorganisms to other items of textiles in the wash. However, if we use stats, it would look like we had a 99.9 to 99.99% reduction in microorganisms. But what happens to those 1,000 remaining cells that are there? If you wash at 60, like the policy states, there isn't an issue. We didn't find any survival, we didn't find any cross-contamination. But how much is this adhered to? The other thing that we have looked at is the survival of C. difficile spores. Now, C. difficile spores, by their nature, behave very differently to micro-vegetative cells that we find. And what we did here was we washed it to the guidelines of the industrial washing, because this was based around bed sheets. So we were looking at the contamination of bed sheets with C. difficile spores and what happened if these went through industrial washing. We used both a type strain, and we also used a strain that we had isolated from a C. difficile patient's bed sheet. And we washed it at 71, and we washed it with industrial detergent and without industrial detergent. And what you can see here, whoops, sorry, let's go back. There we go. What you can see in the first set of columns is that when no detergent is present, that the reduction in spores is minimal. And that also, again, you get a big crossover of those spores onto sterile swatches. If you start adding detergent in, this seems to have the most significant impact on the spores itself. And in the second set of columns, so this is the type strain that we're looking at, so this is, isn't the clinical isolate, you are seeing very minimal amounts of spores surviving. However, the infective dose of C. difficile spores is very low. So although we're seeing a significant reduction, they are still surviving. We then looked to see if the spores from an actual patient behaved any differently. And this was ribotype 001/072. And we saw exactly the same pattern. We saw slightly increased survival when we used detergent, but nothing significant. And this is through high temperature washing, industrial washing, with industrial detergents. What happens if there is C. difficile spores on nurses' uniforms being washed at 40 degrees? And here we have some of those spores in those fibres surviving. So you can see them bundled together. The second image is a bit up close, but you can see just a few cells there. But when we've zoomed out a little, this is using SEM, so scanning electron microscopy. You can see that there's quite a few cells embedded within those fibres. Something else we also found that was very interesting was when the laundry is collected off of your wards, it goes into a sluice room. That's then removed to usually an outside area where the launderers come and collect them from. We found that over a 48-hour period of time that the C. difficile spores grow some type of anchor, and the removal of those spores from the textile at 0 hours compared to 48 hours decreases by up to 40%. So I think, again, it illustrates the need for more immediate washing of textiles. So what's the risk associated with this set of data then? Monitoring adherence to policies. Who checks the policies? Who checks the nurses are adhering to those policies? How much knowledge transfer is there? How do nurses go from one trust to another trust and know how they should be washing their uniforms? Because the policies vary. Some of the most used temperatures are 40 degrees in the domestic setting. Does not kill all of the bacteria, and there is a transfer to other sterile items in the wash, meaning they can become contaminated. Is there a link between these textiles going back onto the ward and infecting patients, the environment? That's a question that we still need to answer. So we've published all of this work, and I wrote an article for Nursing Times. Some of you may subscribe to that. And the article was in this month's journal, and it's also part of their journal club associated with the CPD points for this month. So if you do get that, you should be able to pull that article up, and also the journal club questions that I stated around that for discussion. And there's just some of the acknowledgements of the funders and the people we've been working with. And now I'm going to hand you over to Val.
Thank you, Ian and Katie, for those presentations. Ian left with a question, is there a cause for concern? And I think the way we're going forward, yes, there is. And one thing Katie didn't touch on that I thought she might have done was talking about biofilms in washing machines, which at the moment we do know is a huge problem. So let's look at infection prevention and the problems and concerns we could have in our healthcare environments. We know healthcare-associated infection is an issue. We try to prevent that as much as possible. And if anyone ever has anyone in their life who dies of a healthcare-associated infection, you are extremely angry with the healthcare system. There's a growing concern of antimicrobial resistance. That's all we're talking about today, really. We've had Jim O'Neill looking into it. What's interesting is that we've started to look more closely at the role of the environment in infection prevention. I think with Wash Your Hands campaign, it's demonstrated that if we do have an intervention policy, that we can make a difference to infection rates. And perhaps if we do clear the environment where our patients are staying, that we can make a difference. So what about prevention of infection through textile contamination? Ian mentioned a few. Katie's demonstrated that it's possible. But how much do you really know about it in your healthcare environment? I want to ask just a question whilst we're here. I know they've started to remove a lot of the dryers in the hospitals, but you're there, you stood in your uniform, you wash your hands, yeah? You do it 2 minutes and you sing your songs. And then your hands, you dry them, you shove them in the dryers and they blow all over the place. But then your hands are still not quite dry. What do you do with them? What do you do with them today? You do that, don't you? And that's where we find most of the bugs on uniforms, in pens and pockets. Fascinating. And what I want to end the talk with today is talking about incorporation of biocides, including silver, into textiles and plastics, and what effect that may have in infection prevention. So it was 2011 when Margaret Chan first stood up and said things as small as an infected scratch or a sore throat could kill people. And certainly we've seen it. It's been never out of the press with sepsis over Christmas. It's been very, very high. But it's also been estimated that the current antibiotics we've got, if we don't do what the stewards are telling us we have to do, they may become useless within the next 2 decades, and that's terrifying because we've lived with antibiotics. I remember my mum was one of the first people who got given penicillin when she had diphtheria when she was a young lady. You know, she survived till she was 82, and certainly I think the majority of us would like to have antibiotic cover. And what certainly we may advocate in the future going forward, if the tight controls don't prevent healthcare infections, is we might have to do something even more serious, and we might have to withhold antibiotics and keep biocides and some of the alternatives for more superficial infections and also for prevention. We've got the Jim O'Neill report, and this is an absolute read, honestly. It's fascinating to read, but we've actually reached a critical point. What's not mentioned in any of these reports really is the impact of linen and uniforms, and why we use uniforms, and why have we always done laundry in a certain way to clean things up and to remove and reduce bacterial numbers. There's 10 key areas in Jim O'Neill's report, and you can see them there. They're very well listed, but the one where textiles should have a role to play is one of the second areas where you're talking about improving hygiene and preventing the spread of infection. Now, for years and years and years, and I'm old enough to remember the old post tubs, and I remember my mum with the post tubs and the boilers to boil up the temperature of the laundry, and we used to have to do some minging things actually with laundry when I think about it. But we've all now gone into the automatic washing machines, and I don't know about yourself, fascinated by Katie's report, but I would like to actually challenge you here in the room and ask, has anybody ever washed their uniforms with the rest of the laundry? Yeah, put your hand up, it doesn't matter. Yes, I think the majority of people do. What do we do now? We have colour-fast colours, whites and towels, which we might wash at 60. Occasionally we do a 90, but my God, it takes forever in the washing machine, and it costs so much electric. And then you have the quick wash at 28 minutes at about 30 degrees C with a bit of washing powder. And actually, the reason we launder in the main is to clean our clothes, is to make sure they look clean. We've not always in the home been worried about getting rid of microorganisms, because actually if we live in a sterile environment, we've also got major problems for the future as well. So it is a bit of a concern that you're bringing healthcare associated organisms home and putting them in your laundry. Yeah, so it could mean that your knickers are contaminated with a bit of C. diff and MRSA, or your husband's shirt has something nicely down his sleeve. And what else are we doing with laundry? We don't like to iron anymore, do we? So, you know, tumble dryers are good because they get nice and hot, but if you're putting it over the maiden and then you're not ironing, so other factors that actually help reduce organism numbers are being taken away, and we're relying solely on laundry. We've got some shocking statistics with healthcare associated infection if we don't change. And if linen does have a role to play, we need to act on it very quickly, and we need to know those figures. So they reckon that 1 person every 3 seconds will be dying of a healthcare associated infection in the future if we don't have antibiotics. And that AMR in 2050, there'll be 10 million deaths, higher than cancer. And if you think about it, we've all been dying with infection all our lives, but we've had antibiotics. It's been brilliant. So we have got some very shocking figures going on in healthcare associated infections in healthcare environments. And I think the one area that we don't look at is our linen and our textiles. And if we can do something to reduce infection rate, I think we should, and I think it's important that we look at it. We know microorganisms are shed into the environment very high frequency with aerosols and touch. We've done all that work in infection prevention. We know that in this room, these carpets will be full of spores and gram-positive organisms. If we go into the toilets and bathrooms, we know there's going to be predominance of gram-negative organisms. Nebulizers, everything. But we know linen and uniforms come contaminated with our hand washing and healthcare environments. and they can cause subsequent cross-infection, but we don't know statistics very well because linen's not often seen as a focus of infection. Now, what do we know about biocides and biocide incorporation? We've had some very interesting studies. I'm only going to focus on silver, although there are other biocides available, and I'm going to talk about silver. But we can incorporate them into a variety of medical devices, and we already have done, to prevent the need for antibiotics and treatment and to prevent infection. There was a really big study done associated with ET tubes called the Nascent study, where they incorporated silver into the plastic, and they saw a massive reduction in the number of ventilated-associated infections. Silver impregnated dressings. We must all be used to seeing these now and other biocides in dressings. We are using them to try and reduce the number of antibiotics and to treat infection. And we know that if you incorporate silver into dressings, we can actually treat infection without antibiotics. Now, incorporating silver into textiles is not about treating an infection. It's about preventing an infection or a potential infection. Okay? Could silver impregnated textiles actually help reduce infections associated with linen? What do we know about silver? It's been used since antiquity, the silver coins in the fountain and everything else. But it's not metallic silver that's antimicrobial, it's silver ions that are antimicrobial. And when silver ions are in a moist environment, they are highly antimicrobial. There is 4 different mechanisms of action, unlike an antibiotic. So, for example, penicillin affects a bacterial cell wall, gentamicin affects protein synthesis at the ribosome level, whereas silver attacks cell membrane, it integrates with DNA, it integrates with the proteins in the cell cytoplasm, and it also interferes with exchange at the cytoplasmic membrane level. So it has 4 different mechanisms of action within a cell. So using silver to kill a bacterial cell, it's a bit like having a knife, a gun, and a hangman's noose. So it's got 3 or 4 different modes of action. It's highly antimicrobial at 12 to 50 parts per million. That's very, very small. It's about 0.012%, very low levels of silver ions. And the quantity of silver ions in the textile can vary depending upon about the polyester component. The silver ions that are usually incorporated into the polyester during production. So depending on how much polyester you put in a textile, you can alter the level of silver activity. And really just to show that there have been a number of studies on BioShield undertaken now, and I certainly work in a laboratory that does the work for them in the UK. And we've shown that silver ions incorporated into a variety of fabrics, towels, sheeting, pillowcases, and uniforms can reduce bacterial load by greater than 3 log. Now, I've said greater than 3 log, so if it had 1 million cells on it, it would take it down to 1,000 cells. And I don't want to get into inoculating dose, etc., but it can reduce drastically. That is international standards. However, with the work that we have been done, we've been adding around about 10 million cells to a swatch and undertaking the standard. And what we've seen is we've seen a 7 log reduction within the space of 24 hours. Now, if you've got textiles that are being stored, ready for laundering, it means that the microbial load can be reduced. And also, it is possible that we've actually seen, and there's some work just come in now which has changed since I've had this slide made. We had data to show that if you had swatches and you washed them 100 times, that the antimicrobial action was still there. Because it's one of the questions you get asked. Is that antimicrobial action going to remain after washing, or is the silver going to leach out? The silver doesn't leach out, it remains in the polyester. But the latest information we have is that it survives 400 washings, so it means the longevity of uniform wear will increase. So certainly with the data I've seen in the lab, we've seen that silver in textiles could possibly help reduce cross-infection in healthcare environments by reducing the bacterial load during storage, but also after laundry. So, thank you very much.